Thanks very much. So I want to start for people can line up for questions, but I had a few questions of my own for the group. First, we've heard over the last couple of days a number of different concerns raised about data quality, about the nature of the databases, about the software methods used in analysis and interpretation of oncology patient data. How do we ensure the computational methods and tools are appropriately validated, and what sort of oversight mechanisms do we need to do that, whether those are regulatory mechanisms or non-regulatory mechanisms? What kind of oversight do we need to make sure things are validated? Lisa?
Well, I can tell you one thing that works quite well. It's to put it in a clinical trial where you have an honest broker in the form of a statistician or computational specialist who is the one who can make the link to the outcome data. And if you have everything pre-specified and somebody's actually even got the code, even if they can't look inside it, somebody's got the code, you plug it in and you see if it works. In the end, there's nothing like that to make a convincing argument, my experience. And I think that was one of the reasons for success of the Oncotype DX story. I mean, they, from the very beginning, had it set up to have an honest broker and carefully defined what they wanted their classifier to do, what the criteria for success would be, and it just, it was a happy ending to that story. Now, sometimes it's not going to be a happy ending, but at least you know what the ending is.
Okay, other thoughts? Yeah, go ahead.
To add one thought to that, when we talk about validation, I think we sometimes lose sight of the clinical context or the intended use. There's lots of classifiers, there's lots of algorithms, there's lots of genomic tests, and I think it has to start with what is the purpose of the test. If I'm a developer, why am I developing it? What do I expect people to do with that information? And that, it seems to me, is often lost sight of. People say, I've got a great technology and it can be used for a million things. Well, what should it be used for? Because unless you can declare that up front, then it makes it very difficult to design the appropriate validation studies because you have to validate against an intended use. So I just sort of put that out as a plea to our community to keep in mind that it has to start with, what is this test supposed to be good for?
Yeah, I want to strongly second that. I mean, especially in the early days and even now, we see tons of predictors out there in the literature. You get a significant p-value because you can separate two survival curves, and you're scratching your head at the end thinking, well, what the heck am I ever going to use this for? And again, I think not to pump up too much the Oncotype DX, but that was, again, one of the things that I think was part of their success, that they had a very carefully defined question. So we have these women with early stage breast cancer, hormone therapy, and which of those patients really need chemotherapy? It had a very nicely defined question. Now, all of that said, the TAILOR-X trial accrued over 11,000 patients. We cannot do that for every single test that comes around. So that's why we're going to have some very high quality databases, specimen banks, so we can do some of these in very carefully designed, what is often referred to as prospective retrospective studies. You also find out pretty quickly when you attempt to do something like that even though we say, well, we have banks full of specimens, when you actually start trying to answer a carefully defined clinical question in a particular patient population, you soon find you do not have as much data or specimens as you think that are really relevant to that particular question.
So, yeah.
So I just want to second that. I think we need very significant scale as far as data goes. I think the examples we heard, Oncotype DX, are the one I used, where you have a very specific indication, very specific drug combination. That's what we have right now. But I think eventually we will have switched to a disease-specific decision support, and then it will get really complicated, and that's when we need the scale data. So just a prospective phase 3, or even if it's a large cohort like the Oncotype DX, probably isn't going to be big enough. It probably has to be significant real-world data in combination with the clinical trials and so forth, right? Yeah.
I think Bray and then Frank.
Yeah, I think we have issues in quality around really trying to understand, are these data sets reflective of the intended population? And so in clinical trials, the FDA does a great job of saying, let's take a look at this, but I wonder about the diagnostics and the analytics it's hard to necessarily know exactly, who was in the data set. And I think what, was just being said about if we don't even know what the intended use is going to be, it's hard to really define, was this a high quality data set that was appropriate for this technology? So I think we have work to do there.
Maybe I add, one of the opportunities that I see practically are platform trials and pragmatic trials with master protocols that can be historically followed through. And I think it's not just for therapeutic areas, it's also for in vitro diagnostic and test development, because there your population and the, let's say, the history of the disease is documented within the same system. And some of the issues that we are struggling with is, of course, the difference between standard of care, electronic health record documentation and information, and the information that's collected in clinical trials. And I think that's an opportunity to merge it, number one. Number two, there's the opportunity to include several innovator drugs or treatments, and there's also the opportunity to there's space for innovators in terms of the test methodologies and new test methods. So I think that's something we need to explore and continue to.
So Lisa's point at the end that she made about the importance of then having really good data sets that we can use in different ways to really be able to make this work and to be able to create the research that would validate all this sort of stuff really hinges to some extent on the ability to have robust approaches to data sharing, right? So I guess the next question I would ask is how do we create an environment that really incentivizes that kind of collaboration to really make that possible? What infrastructure and resources are needed? How do we remove barriers and disincentives to that sharing?
Well, obviously, NIH has a big stick, which is that it's parts of your grant award terms. But even with that, we have difficulties sometimes because of proprietary issues, because of patient consent issues, and so it's not, snap your fingers and it's done kind of thing. But I think that the community is going to have to really come to grips with particularly the consent issues. I mean, we have a lot of trouble with that. If it's genomic data, depending on the IRB that looks at the proposal, they may or may not be happy with making the data shareable, or may put so many strings on it that it's virtually impossible, or could take you 5 years to ever get the data once you apply. So I think we really have to develop some better systems for how we handle that. And I do, it's interesting. I sometimes speak with patient advocate groups as well, and on the one hand, we hear some of the regulatory bodies or the IRB saying, the patients don't want their data out there. I talk to the patient advocates and they say, we're dying to give our data to people. We want you to use it. So there's still a very big disconnect there. And I don't know what the solution is, but we need to work on it.
Yeah, go ahead. So actually, Lee and then Devin.
It's probably worth looking across the ocean at the UK, where they're now doing this kind of work routinely on literally hundreds of thousands of Britons. And it took a uniform governmental approach to make sure that data was accessible, but it is. And the citizenry of the UK is not rising up and saying we feel violated or that our data is being used in wrong ways. But it took national consensus. It took the Prime Minister saying this is something we should do. And I think it might be a little hard to get that kind of attention in American politics, but if we could, I think you'd find the kinds of answers you were already talking about, Lisa, and that people want their data used to further good health.
Okay. Deb?
Yeah, I mean, I really referred to this both in my presentation yesterday and in the moments that I had to speak at the beginning of this session, which is, to sort of have certain reuses of data be considered to be routine, subject to certain conditions. But it also, look, when patients have the data and can move it to where they want to, that's yet another answer. It's not the only answer, but it's an answer that's completely underexplored today. Patients have the right to their data under HIPAA. Patients have the right to their data under GDPR and also under the new California law, but the institutions are not well situated to easily provide it. But certainly in terms of the patient groups that we talk to, we hear the exact same thing, Lisa, which is use my data to help me. use my data to help others like me responsibly. So I think we're hearing some common themes that I think we can pull on and figure out ways to make that work.
Yeah, and again, building on the examples of the UK or the Scandinavian countries, there is a clear indication that there are significant advantages to this ability to access and utilize public health data. Now, in our own situation here, I believe the law would allow, first of all, the patient to contribute the information, and that includes even clinical trials. And as you know, also the patient-level data that is submitted to the FDA is public information as soon as the product is on the market or the product has obtained approval. Now, for the phase in between, where it's under review, we are still dealing with some legal interpretation, yet I would say from a purely legal perspective, I'm not a lawyer, but I could stipulate that Title 18, Section 1001 could be interpreted in such way that anything in a government process, and that includes, of course, patient data, under review would be government process, is public information and therefore cannot be concealed. But again, to enforce that and change the culture, there are different ways of doing it, and maybe industry also realizes that there's an opportunity to move ahead and voluntarily open such kind of information for the public and the public good.
I guess I wanted to ask Devin a question to ask you to follow up on a point you made about. So yes, under HIPAA, patients have a right to request their data, but providers don't necessarily have a requirement to provide it in a way that patients can use it. And so, how do we get to the point of, if I go to my doctor and say I want a copy of my medical record, and they print off a PDF of everything that's in Epic, it's really not too useful to me or anybody else. So how do we get to the point of the provision of the data being in a format that's actually useful?
I think there are a couple of things going on. It's a really good point. There are companies out there that are actually that are figuring out ways to pull data out of PDFs. But in the meantime, all of that work would not be necessary if the data were provided in a portable manner. And the GDPR actually does require that the right of the individual to obtain their data it has to be portable. That's not really as clear in HIPAA. There's, you have to put it in the form or the format that the patient requests, but it has to be readily producible in that form or format, and everyone is essentially defaulted to PDF, which is not it's definitely not as usable. But there are many things that we could do, at least from a U.S.-centric perspective. There are standards that are set for the electronic health records that get certified by the HHS, Office of the National Coordinator, in terms of data that's affirmatively made available to patients via portals or soon via application programming interfaces or APIs, where that data has to be in certain formats and it has to include certain data elements. It's not everything. It's not nearly enough. But they're trying to move the ball towards a reusable data set that the patient can take and port around for care purposes, but also ultimately for research. Lots more work to be done on that. But I think the ball's moving in the right direction. It's just moving incredibly slowly.
Anybody else on this? Okay, so we got a queue.
Yeah. Hi, I'm Weijin Ko. I joined NCCN 6 weeks ago. I do have two questions for the panel here. It seems to me that the title of this workshop is not completely accurate. I do get that precision oncology improves cancer characterization and care. As an oncologist, when patients come to see me, 99% of the time I know they have a cancer. Can this computational methodology, machine learning, AI, be moved upstream to help primary care physicians in the diagnosis of cancers before we see them at a stage 3 or stage 4? That's the first question. The second question is, how do we involve or change the physician-patient construct? What I'm saying is that precision oncology, I totally accept, is better treatment. You choose better options, but it is not synonymous with absolute effectiveness. And at this point in time, when we tell patients that this is precision oncology, and we understand the numeracy and the literacy gaps in our population at large, how do we educate patients that, well, this is better? We realize that this is not an absolute, especially if the choice that we made that is helped by machine learning is perhaps initially counterintuitive.
So I can respond to the second part of that about the literacy and the numeracy. I don't know how many of you turn into grand rounds for NIH Collaboratory and PCORNet every Friday, 1 p.m., open to the public, and all the webinars are recorded and stored on the website for replay. Last week, we had Brian Ziekman Fisher from University of Michigan talking about how we can better communicate physicians and patients, talking about the risks and how much people are likely to benefit and potentially not, and how you present that information. It's really complex information that needs to be simplified, and there's not a good, easy answer. But I think if you want to go back and watch that webinar, he presented some scales and things that can actually help support those conversations.
I guess to comment on each of your questions, the second one first, to some extent, I think we, the oncology community, are very much to blame for much of the so-called hype about precision medicine. And we're to blame in part because much of what we talk about and report on and what we discuss with the lay press are the exceptional responders. Even using that name, has created a certain veneer about precision oncology that everybody's going to be an exceptional responder. And of course, it's the needle in the haystack that's the exceptional responder. And I think we also have a real challenge in sort of describing the application of precision oncology in the relevant clinical context. I think everybody would agree that there are many examples, where at the time of diagnosis of an advanced cancer, that having a genomic test that informs a treatment decision with an FDA-approved drug that's proven safe and effective, that's a good thing. Where we begin to delve into the, as Lee put it, the off-evidence space, is in the advanced cancer patients where we're doing these comprehensive genomic panels, which oftentimes reveal treatment options that are not of any proven effectiveness in treating a patient. And yet we sort of create this idea that if we can only find the variant that we can target, that somehow there'll be a miraculous outcome. And of course, we know the data doesn't support that. So I think we have to take some responsibility as a community for setting the record straight on where we are in precision oncology, and conveying that clearly to patients. I think on the issue of, where do things like computational methods help at the diagnostic end, I'm certainly no expert in that, but it seems to me that, one of the places, of course, will be where Hedi Hrisak talked about its application in radiology and, helping to determine signal to noise. I mean, things like lung cancer screening, we know that the majority of nodules that are picked up on screening CT scans are benign. How do we can't biopsy every single nodule. How does the radiologist and the primary care physician determine, is there really an at-risk lesion there? Should it be biopsied? When should it be biopsied? So I think there are ways that we can move these computational methods earlier into cancer diagnosis, but we have to first ask the question of, what is the purpose of doing so? What is the clinical problem we're trying to solve?
Probably also mentioned in the early screening, there are obviously a lot of companies looking for circulating free DNA to look for non-detectable cancers or early stage, and that work is proving to be a little formidable, but it still holds promise, and that would clearly be the desired object of all of us, is to not have a stage 4 cancer patient. I did want to comment briefly on breast tomosynthesis, computational method to improve mammography. And looking at just raw claims data, which obviously has lots of issues. We had about 200,000 patients in a year who had received breast tomography versus about 1 million who had routine mammography. And after screening out all the people who went back for multiple exams, we found basically that we spent $250 million additional dollars to have no change whatsoever in the callback rate for false positives. And we did increase detection rate by 0.2%. So there's a question there of, okay, yes, that's a positive result, but is it worth the value? And I'd love to see the clinical study that said to those 0.2% of women were those really kind of benign, overdiagnosed women versus true cancers where it made a difference. So I think if we move up the scale, we have to be awfully rigorous about our enthusiasm for technology to make sure that it really works and that it offers some value. Because again, there are going to be limited resources in the next decade.
So let me piggyback on that comment. I was going to start by thanking everyone who's touched on the various aspects of precision oncology in the space of primary prevention. And maybe make the point that what you just said is even more important if we move even further up and try to identify strata at high risk, not just in an early detection setting, but in a primary prevention space. There, the potential is enormous, I believe, for machine learning to identify people at high risk, motivate behavior change, help them understand genetic information, for example, and the list could go on. But the challenges are also magnified by the breadth of the populations that need to be involved. So I hope as we continue our policy discussion about precision oncology that we don't leave behind the precision prevention side.
The caveat that we understand how a human brain works. I mean, I'm an oncologist with 6 kids, and one of them is a smoker. And you can imagine how I feel about that and how much pressure, but I wish I understood what the right button was to press to get him to stop.
Great. I'm Pratik Shah. I work at MIT in Cambridge, Massachusetts. Thanks for having me here. It's great. I have some comments and open questions to the panel. So I think in the computer science world, we are seeing that there are large data sets being created by computational methods. They often live in silos. We also see that large investment is going to happen in ML, machine learning, and AI, like millions of dollars. And that's going to lead to like new evidence created by these methods. Whether we like it or we don't like it, the money is coming, evidence will be created. And most of it would live in an academic graveyard if there are no bridges between medicine and computer science. Right, so that's like one key observation. I just want to put it out there. That's happening. There's some requests. So one is like the FDA people are here. Please clarify to us when we do machine learning research, what regulations do you expect? Like what is the regulatory pathway for algorithms to be vetted out? And I know that's ongoing conversation, but the sooner we move towards that, the more the money that's being pumped into this field will be used for public good. That's one. Please let us know problem statements, data sets, and your expectations that you have from the computer science world. We are happy to work with people in oncology, infectious diseases, what have you. Once we know where your data is, what your questions are, and how do we go after the problem that benefits patients. So we are driven by the same motives everybody else is driven by here. And I think one of the key recommendations to the National Cancer Forum would be to create bridges to the computational world. And I'm happy to be a small bridge. I don't know how big I am, but like we can help and kind of bring these conversations to the computer science world. So, and that's something I would love to hear from you. How do we create these bridges? And in my personal research in my group, what we are going after are pragmatic clinical trials. We believe that is patient-centric medicine, and we're going to throw machine learning, whatever we want, to make that happen. And the omics data sets are another huge opportunity where humans cannot process them, because they're so large and correlations and causalities are not known. So we'd love to create some sort of conversation around these things. Thanks.
Does our representative from the FDA want to?
Let me take a first step. So first of all, I think what's really critical in this process, we have to understand it as an iterative process, a learning process. So let's keep it transparent. But let's also consider things like training sets and data sets that are actually of a certain quality that lend itself to common use. That's historically a problem, but I think that's something we will come to. As you pointed out, I think one of the options, or we call this regulatory science, or science where actually FDA scientists are involved in the science that's being done, and that's a active field of expansion. We have right now 4 centers of regulatory science and innovation with academic centers. I think that's one of the platforms that we try to expand and leverage in this dialogue.
So I would say there's real opportunity to develop patient advisory groups for your work. It is true that computer scientists and engineers are separate from really care and access to patient perspectives regularly. And so there's a lot of opportunity. The oncology community itself benefits from having some of the most skilled and knowledgeable advocates around. And so I think you would find if you set up these mechanisms, you'd have willing partners who would participate. And we've even seen models, aside from, design of clinical trials, working with patient groups. I know that there are actually tumor boards that have patients participating and at the table. And so I think we just have to look across all of our activities that we're calling patient and people-centric and think about how we really engage patients in them.
Can I make maybe a very short comment? I think another one is that actually folks of your discipline start working or even getting their hands wet in the healthcare industry. So we, for example, we have huge issues hiring folks with AI experience in the Bay Area. We actually, Roche Diagnostics, actually, they had to put out ads on the 101 because folks didn't even know who Roche is or what healthcare is. So there's sort of that's another area. Also internally, we are running competitions with different data sets where 2 or 3 AI companies from the outside compete against an inside team. So we're really just trying to get the disciplines in contact. But I think we both have to learn from each other. My experience is with data. I have a data analytics group also in the biomarker section. We're trying to integrate things. And I think the analytics folks probably have to learn healthcare or oncology a bit more rather than the other way around, because it's easier for you guys to learn what we do than the other way around. That's sort of my recommendation, I guess, for the bridge. Yeah.
Specifically to the question of tomosynthesis that you mentioned, we have started a study, a randomized study of outcomes comparing tomosynthesis to digital mammography. It's called TIMMIST, and it has 165,000 sample size. So that gets actually to my main point. That is, for most of the diagnostics, for the 99.9% of all the new diagnostics that are being discovered, it is not obvious what's the business case. Absolutely not. And the money that is driving some of that development, A, is NIH funding right now. B is venture capital. A lot of that comes with very strong strings attached, especially the venture capital, and it will go away very quickly if it doesn't see that there is adequate, there's real prospect here for quick return. So I don't see how this is going to be funded in the long term. In particular, we're talking about a translational, research program in these diagnostics to bring them to the bedside. Who is going to fund this is absolutely not clear, because it's absolutely not clear who will pay most of it. Tomosynthesis you will pay anyway if it comes along, because it's another kind of mammography, et cetera, et cetera. And if it doesn't show that it has a real impact, then maybe people will walk away from it. But it's a big enough ticket. For most of the things that we're talking about, they're not big enough tickets, and the development of them costs a lot of money, especially in the diagnostic space where it's not saturated with investment that way. So I really don't know how this will all work. This is important. I mean, it's less relevant. It's less so in the biologic space, because for some reason the drug companies are also closely related to this. But in the diagnostic space, it's a big question. It has to do with how are we going to come up with a
an important sort of a doable translational pathway. And one situation where the whole thing comes crashing down is when you say, show me what you get, what's the bang for the buck? That's a very tall order if you want to show how the particular diagnostic affects outcomes, patient outcomes. And we run this routinely, even with large-scale diagnostics, but the smaller-scale ones, unless somehow they're incorporated into the practice patterns within, say, hospitals or within care providers, and they become part of a bundle, I don't see how else anybody will be able to move ahead and develop these.
I'll jump on that just because I think you hit on something so important, which is what's most important is patient outcomes. And at Biden Cancer Summit, we had a breakout session where we came up with all kinds of great ideas. And then people said, yeah, but I've got this MBA who doesn't know anything about what I do or the value, who's measuring things and making decisions for our organization and what I can actually implement, that the patient interest might not be what's at the top. And so I think we have to have some real transparency around those things that are holding us back.
So it's the only silver lining I can think of for drug pricing, but it does make an economic model for better diagnostics, because either spending $100,000 on the right drug or avoiding $100,000, those kinds of diagnostic biomarkers in particular, I think there's still a very good economic case that would promote funding and doing the research. But for other issues, I think you're absolutely right. Now, the other piece of this is even a validated test has to be used correctly in the field. And we talked about Oncotype DX, but I spent the first year of its initiation, I think we had well over 600 to 700 denials of clinicians using it in totally inappropriate circumstances. So there is yet that second phase of they had good evidence, but the evidence was quickly being ignored or expanded into what I call the off-evidence area, and it really devalued the test in the minds of people who had to pay for it. They forgot about how good it was in one area, but not in others.
Okay. I think this will be the last question.
Last question. And so I know this will be the weird question, but that's okay. We've been focused much on the individual, but this is going to come from a population health sort of perspective. If it's true that the ZNA, that is the zip code in which you live from, actually probably has much more of a predictive outcome of your health than your DNA, what I was thinking about is how do you sort of foresee computational sort of methodology in the context of analyzing sort of communities? I'll give you a quick example. Someone's living near a highway near a diesel fuel, or near living in an area where there's high levels of incidence and death, blah, 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 from a certain particular cancer because of arsenic. How are you actually looking at that exposome, those data, in your computational analysis, just to dovetail on the prevention? Because frequently we look at the individual, but we never really link that individual to really an area in which they live in. So it's just a question. I know I'm a pain in the butt, so I'm sorry.
Well, if I may, certainly there is high awareness of your pointing towards the specific living conditions and environmental factors and other cross-cutting data that is important and impacts our daily life and certainly our wellness and when we project futures. And this information has to be integrated in our assessment of health and disease, and it should become a factor. But again, it is going back to the reliability and the density of the data that's available and the potential impact that it can make. And we are at a relatively early state, some of the digital revolution, and we were trying to master some of the very basics at the agency. And of course, it can be stipulated that it'll take a while. Some of the assessment of innovative technology from discovery or first description to making a product takes between 25 and 37 years. And you all can think of specific examples, I know. That's just reality right now, but I think we can change that. So let's be optimistic in terms of the ability to work through sizable data sets, integrate them, make them interoperable, and create models that inform each other with the different components. And I think that's where we need to go.
So I'm going to take the prerogative to maybe make a last point about that. The All of Us campaign is doing a great job, I think, of collecting some of the data for the social determinants of health. But I think what's an open question will be very interesting. We'll see the actual uses of that data. So when genomic data is available, social determinants of health data are available, what are researchers going to really be focused on and gravitating to, even if we know that the social determinants of health are the primary determinants? Not clear that that's going to be where the excitement and action is. So I think your point is very well taken. And I think with that, we're going to maybe thank all of our panelists and for good job. Thank you.